Hi. Now we're going to do the third in a series of videos on the innate immune system. This one is on the body's second line of defense. So let's share the screen again. Okay. All right. So this, to continue with the analogy of a house, this happens when there's a break-in. And so the pathogens are getting into the body now. And the second line of defense is composed of many elements, cells, chemicals, processes. But we're going to go to the blood because a lot of them originate in the blood. And so if we look at the typical schematic of the blood, you'll notice that plasma isn't in here, but that's the first thing we have to talk about. The plasma has mostly water and some electrolytes, dissolved gases, nutrients. And then the serum is the fluid when the clotting factors are removed. And plasma also contains these iron binding compounds because iron is needed for metabolism. And these iron binding compounds, one of them being transferrin, a protein, will sequester the iron to keep it away from the microbes. Iron is essential to us for the electron transport system. It's a cofactor in many enzymes, and it's also needed in hemoglobin to carry oxygen around our body. Also in the plasma, we're going to find the complement proteins that we talked about before, and then antibodies that we're going to be talking about in chapter 16 when we talk about the adaptive immune system. So let's look here at the blood, and you see that there are defensive components of blood called leukocytes. Sometimes we think of these as white blood cells. The cells and the fragments are called elements. And the formed elements are erythrocytes. So you can see those here. Platelets, and the platelets are considered here. And then there are leukocytes, and the rest of these are the leukocytes. They're involved in defending the body against invaders. And you can see that they're divided according to what they look like. The granulocytes, where you can see kind of granules inside these cells, and the agranulocytes where you don't. And you've seen this depiction before, probably in a prerequisite course or in A&P. Here you see a blood stem cell. So this is another one of those adult stem cells that we were talking about earlier, okay? Adult stem cells are different from the embryonic stem cells, and the blood stem cells in bone marrow is what makes the bone marrow transplant a valuable tool in combating certain diseases and cancers. When we look at the leukocytes, here we've got some of the granulocytes in stained blood smears. Okay, so this is what they actually look like under the light microscope, and you could see them there. The granulocytes have three types: the basophils, these are the basophils, they stain blue, and eosinophils, they stain red orange, but you can see that they've got the blue as the nucleus here, and then the neutrophils, they stain lilac with a mix of basic and acidic dyes. In all cases, the nucleus is seen as blue, and you can see that the nucleus is lobed. Okay, these are unusual nuclei appearances. The neutrophils and the eosinophils, okay, the neutrophils and the eosinophils are phagocytes, and they're going to phagocytize pathogens. They're also capable of something that we call diapedesis, and that is where they can squeeze between the capillary cells, so they can move around that way. The basophils don't phagocytize. However, they release inflammatory chemicals, and when we talk about inflammation in a little bit, we will look at that. When we look at the agranulocytes here, okay, You can see that this has, the monocyte has a lobe nucleus here. And the lymphocyte, the nucleus takes up almost the entire cell. The lymphocytes are the most common involved in adaptive immunity. So, we are going to be looking at these further when we talk about adaptive immunity in chapter 16. But the natural killer in lymphocytes are part of innate immunity. The monocytes here are going to leave the blood and mature into something called macrophages. And these are going to be phagocytic cells that would dissolve the foreign materials, or devour them rather. Okay, so the leukocytes are defensive blood cells. So let's go back here. And these are the leukocytes, the defensive blood cells, all right? Sometimes known as white blood cells. So we can look at a differential white blood count and can signal disease. So if we look at the differential, like how many of each of these are around, you'll see that if there are increased eosinophils, that indicates allergies or a parasitic worm infection. Bacterial diseases often show increase in leukocytes and neutrophils, whereas viral infections will show an increase in the lymphocytes. So we look here at the relative numbers of these and can get a handle on the type of disease that an individual might have. Okay, we talk about cells that we're going to phagocytize. So cells capable of these are called phagocytes. We don't really completely understand it. There are 6 stages to it. And let's do an overview here.
Phagocytosis is the ingestion of a solid, such as a microorganism or cellular debris, by a eukaryotic cell. Phagocytosis is used by some protozoans for obtaining nutrients, but this process is also used by certain cells of the immune system to fight infection. These immune system cells include macrophages and neutrophils and are collectively called phagocytes.
Now let's look at the mechanism.
Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system. The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization, a process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome. The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes. After phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation. Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome.
And this is another events. Now, you'll notice that the video talked about 5 stages, and this talks about 6 stages. And so your book talks about 6 stages: chemotaxis, adhesion, ingestion. Okay, there's the ingestion. Then they talk about maturation. And so we don't see that here. Talks about killing, and it talks about elimination. So instead of maturation, we've got the fusion of a series of vesicles, including the lysosomes, and then killing. So this is where you would have the phagolysosome forming. And so that would, the fusion would be the maturization, and then the killing, and then the elimination. It's useful to have an overview, and then to see how it works. And it does say that this isn't completely understood. And so this is our best guess at what happens. Now, there's also a way that things are killed by non-phagocytosis. One is killing by the eosinophils that attack the parasitic helminths. Those are worms. And so they would secrete toxins that are going to weaken or kill it. And then eosinophilia is often indicative of a helminth infection or allergies. So that means, a lot of eosinophils are going to be around. And the eosinophil mitochondrial DNA and proteins form structures that are going to kill some bacteria. And then there are the natural killer cells. They're lymphocytes, and they secrete toxins onto the surface of virally infected cells. So when you see the natural killer lymphocytes, you've got the viruses. They differ into normal body cells because they have membrane proteins similar to the natural killer cells. And then there are the neutrophils, and the neutrophils are indicative of viruses again, and they can destroy without phagocytosis. They produce chemicals that kill the nearby invaders, and they have extracellular fibers called neutrophil extracellular traps, nets, clever, that will bind and kill bacteria. All right, then there is the non-specific chemical defenses against pathogens with toll-like receptors, TLRs. There are 10 different kinds of these. We're going to look at that in a minute. But these are membrane proteins that are produced by phagocytic cells, phagocytic cells, and they bind pathogen-associated molecular patterns, or PAMPs. These initiate defensive responses. We're going to talk about inflammation in a little bit. And then the next chapter, we're going to talk about adaptive immune responses. And then eventually it results in killing of the invaders or the microbial pathogens by apoptosis. So here is a list of some of the ones that we know about, the toll-like receptors and their natural binding partners. So this is the toll-like receptor, and these are the pathogen-associated molecular pattern microbial molecules. Okay, so you see 1, 2, then 3 is down here, 4, 5, 6, 7, 8, 9, 10. Okay, some of them are in the cytoplasmic membrane, some are there in the phagosome membrane. Okay, so the phagosome membrane, remember, is that ingested endocytosis part in intracellular particle that fuses with a lysosome. Here you've got bacterial lipopeptides and then some proteins in parasites. This is peptidoglycan, lipoteichoic acid, and gram-positive cell walls, and also yeast. Lipid A, which we found in the outer membrane of gram-negative bacteria. Flagellin, or bacterial flagella. Again, bacterial lipopeptides, lipoteichoic acid, and the cell wall of yeast. And then they don't know what it is, but influenza viruses have something on them that the toll-like receptor 10 can glom onto and work. When you look inside the phagosome membrane, here we're talking about the microbial molecules being genetic material. So TLR3, double-stranded RNA that we find in viruses, and then single-stranded RNA, which is used for making proteins, and we find those in bacteria as well as in eukaryotes. And then unmethylated cytosine guanine pairs of viral and bacterial DNA. So these have targets. If it's in the phagosome membrane, the target is going to be genetic material. If it is on the cytoplasmic membrane, it's going to be membrane or cellular wall materials. And then there are non-specific chemical defenses. These non-specific chemical defenses are cytoplasmic membranes that are going to bind these PAMPs again, triggering inflammation that we're going to talk about later, suicide, and then some other responses. And we're not real sure how this works. And we get to the interferons then. You may have heard about interferons. That's because they're non-specific, but they inhibit the spread of viral infections. Although some of them cause the symptoms that are associated with them. And there are two types, type 1, alpha, beta, and then type 2, gamma. So here are the actions of these alpha and beta interferons that we understand. The virus infecting the cell, and here we've got double-stranded RNA is that kind of a virus, okay? And so the double-stranded RNA can get into the nucleus, okay? And the interferon gene then will produce mitochondrial RNA that will eventually make interferon protein molecules that that leave the cell. They're exocytosed. The other thing that happens is viral components will trigger activation of the type 1 genes. Time passes. The ones that are secreted, the interferon alpha and beta are released. They bind to receptors, either on the same cell or on nearby cells. And in the nearby cell, the binding will activate genes for antiviral proteins. And they're inactive, but it makes the mitochondrial RNA and the messenger RNA, and you've got these inactive proteins. Time passes. Here, with the main cell, it's making more viral proteins, more viral particles. Time passes, and the infective cell dies and releases the viruses. The viruses then infect a new cell. However, the inactive antiviral proteins become activated. The viral components are going to activate those. And you see that the active antiviral proteins are going to degrade the mitochondrial DNA, bind to the ribosomes, and stop protein synthesis. And so that's going to prevent the viral replication. So this is how the alpha and beta interferons can eventually stop viral replication. Here you can look at the characteristic of the human interferons, type 1, alpha, type 1 beta, and then type 2 gamma. The principal source, the epithelium, leukocytes, fibroblasts, activated T lymphocytes, and natural killer lymphocytes. We talked about the natural killer ones. We're going to talk about the T lymphocytes in adaptive immunity. Here we've got adaptive immunity again. So here is kind of a segue from the innate to the adaptive immune system. Okay, we're back to complement, and we've already talked about complement. So we're going to move on now to inflammation. I think. Okay, inflammation. This is a non-specific response. You get redness, heat, swelling, and pain. There's acute and chronic. So let's look at an overview.
Damage to the body's tissues through heat, chemicals, sunburn, cuts, or microbial infections triggers a general, nonspecific defensive response called inflammation. Signs and symptoms of inflammation include redness, pain, heat, swelling, and sometimes loss of function. Inflammation has three main functions: to destroy the agent causing injury, to limit the effects of the agent on the rest of the body, and to repair or replace damaged tissue.
And there it's healed. Okay, there's acute and chronic. Acute, quick, short-lived, usually beneficial, and it's important in the second line of defense. And the chronic inflammation is long-lasting, and damage to tissues can cause disease. You've heard a lot about inflammation and about anti-inflammatory agents that you can take, non-steroidal anti-inflammatories like ibuprofen and aspirin. And the internet is replete with all kinds of use these anti-inflammatory foods or avoid these foods that cause inflammation. We're not going to address any of that here. We're just going to talk about the actual process of inflammation. So, what is it? The blood vessels have dilation and increased permeability. Vasodilation is going to produce redness and localized heat, and many chemicals will trigger and promote dilation. Oh, wait a minute. Wrong. There we go. Okay. So here we've got what normal capillaries look like. And here we have an example of what happens when the capillaries expand and allow a lot more blood to get to the affected site. And a lot more liquid also. So you can see the dilated capillaries. Complement also plays a part in here. So here's an example with inflammatory mediators of C3A. And this is a basophil. And C3A will stimulate the basophil to produce inflammatory mediators. On the other hand, a mast cell is triggered by C5A to produce inflammatory mediators. And the inflammatory mediators then are what cause these, the dilation, the opening of the interstitial spaces, and fluid and antimicrobial chemicals can be released into the spaces. Monocytes can squeeze through the spaces, diapedesis, and more fluid can come out. So the presence of bradykinins, prostaglandins, leukotrienes, or histamines will cause the cell lining to pull apart, okay, like here. And it allows the phagocytes to leave the bloodstream and reach the site of the infection, but it causes swelling or edema and pain. Neutrophils and monocytes are delivered to the site of the infection, and they're recruited by chemotactic factors. They attach the receptors on the blood vessels and squeeze between the cells of the vessel wall, entering the site of the infection. It also allows delivery of nutrients and oxygen to the site to facilitate repairs. although some tissues can't be repaired. So let's look at the event an inflammation following a cut and an infection. Here you've got a bacteria entering through a cut, and the cut penetrates the epidermis and the bacteria invade. So here we've got the damaged cells, and they're going to release prostaglandins, leukotrienes, and histamine. The prostaglandins and leukotrienes make the vessel more permeable. Histamine causes the vasodilation, increasing blood flow to the site. Sometimes that causes itching, not just pain. The macrophages and neutrophils are going to squeeze through the walls. The increased permeability allows the antimicrobial chemicals and the clotting proteins to seep into the damaged tissue, but also results in swelling and pressure on nerve endings and pain. Okay. But eventually a blood clot's going to form. More phagocytes migrate to the site and devour the bacteria. There's an accumulation of damaged tissue and leukocytes forming pus. And then undifferentiated stem cells repair the damaged tissue. The blood clot is absorbed or falls off as a scab. So let's look at these steps.
Damaged cells release several chemicals, such as histamines, prostaglandins, and leukotrienes. Certain components of the complement system can also stimulate the release of histamines. One function of these chemicals is to cause vasodilation, an increase in the diameter of arteries and arterioles. Vasodilation results in more blood, and therefore more phagocytes, oxygen, and nutrients being delivered to the site of injury. This increased blood flow results in redness, heat, and some of the pain associated with inflammation. The histamines, prostaglandins, and leukotrienes also have a direct effect on the capillaries in the area of the injury. They cause the walls of the capillaries to open and become leakier. This allows more fluid to leave the capillary and enter the tissue spaces, causing swelling or edema. The increased blood flow delivers phagocytes, such as monocytes and neutrophils, to the scene. The phagocytes stick to the walls of the blood vessels in a process called margination, then squeeze through the gaps between the cells in a process called emigration or diapedesis. Within about an hour of tissue damage, the phagocytes begin to destroy the invading microorganisms by phagocytosis. After engulfing large numbers of microorganisms and damaged tissue, the phagocytes die, forming pus. Other components of the blood lead to the formation of blood clots, which seal off the site of injury. The final stage of inflammation is tissue repair. The increased delivery of nutrients and oxygen from vasodilation aids in this process.
And here is a summary of the chemical mediators of inflammation: the vasodilating chemicals, the chemotactic factors, and substances with both. So here you see some things that we've talked about before in complement, interferons. We haven't really talked very specifically about interleukins, leukotrienes, or platelets. We're going to talk about some of these a little bit more when we talk about the adaptive immune system. Okay, fever. Fever is body temperature over 37 degrees C. You probably know it better as 98.6 degrees Fahrenheit. It results when these pyrogens, and a pyrogen is something that causes a fever, will trigger the hypothalamus, and that increases the body's core temperature. So there are various types of factors that would cause a fever. Bacterial toxins, some cytoplasmic contents of bacteria that are going to be released when the bacteria are split, antibody-antigen complexes, and then pyrogens released by the phagocytes that have phagocytized bacteria. So the exact mechanism is unknown. We've had a few of these with the exact mechanism not known, but here's one theoretical explanation of a production of a fever in response to an infection. So this would be caused by the chemicals secreted by the phagocytes, and they travel in the blood to the brain's hypothalamus. The hypothalamus secretes prostaglandin, and that will reset the hypothalamic thermostat. Then nerve impulses cause shivering, a higher metabolic rate, inhibition of sweating, and vasoconstriction. All of this will cause the body temperature to change. Shivering usually causes it to go lower, but the higher metabolic rate, inhibition of sweating, and vasoconstriction would cause the temperature to go up. These processes increase the body temperature to a point set by the hypothalamic thermostat. It continues as long as the pyrogens are present. And so why is this useful? Well, it enhances the effect of the interferons, and inhibits the growth of some microbes, raises the temperature, and there's always that sweet spot where they can grow. And it might enhance the activities of the phagocytes, the cells of the specific immunity, and then some tissue repair. So we're back here to the table where we started, talking about the first line and the second line of defense. This is a really good summary. The complement kind of bridges both of these. And you'll see the barriers and associated chemicals, the phagocytes, the extracellular killing that are all part of the first line. Complement that bridges first line, second line, and as well as adaptive. And then the second line, the interferons, the antimicrobial peptides, inflammation, and fever. So finally, can you answer this question? Why are pathogen-associated molecular patterns necessary for troll-like receptors to fully function? Think about that. Go back and look at the toll-like receptors, these pathogen-associated molecular patterns, and what they do. And that's it for innate immunity. I will see you in less than 2 weeks. Next Monday, you have an exam. And so you need to start to prepare for that. The exam is going to be online starting Friday night, and it won't be due until next Tuesday. And then next Wednesday, the 27th, the entire school is shut down for a school-wide conference on the future of ACC. And so the next time we will meet in person is going to be April 1st, the day after Easter. I am having surgery on the 20th, and will be out of commission for a few days after that. I'll be checking my emails and things, but I understand that I should take my painkillers before I need them for the first 3 days. So the 20th, the 21st, and the 22nd, probably out of it somewhat. But back on the 23rd, when you'll start taking your exam, I will be responding to texts and can do any Zoom calls if we need to.